Good morning, everyone. Thank you, Ms. Chintana. It's a pleasure to be here today for this session. Actually, I thought I had 30 minutes for the talk, but I'll try to be very, quick now, since I had just have, 10 to 15 minutes. So we all know Nipah virus. It's an emerging issue, not because the virus is new, but because it continues to re-emerge with high fatality and high, public health consequence. So the focus of my presentation is on laboratory capacity and preparedness for Nipah virus, not only during the outbreaks, but also in between the times during those outbreaks. And for regional public health laboratory perspective, preparedness basically it means being able to recognize the threat early, to support the surveillance systems and to basically smoothly integrate into national and regional response frameworks. So this slide basically it summarizes the major Nipah virus outbreaks that have been reported over the period. And you can see starting from the first recognized Nipah virus outbreaks in 1998 in Malaysia and extending to the recent 2026 outbreaks in India and in Bangladesh. One key thing to note here is that you can see that the case fatality rate is very high in these outbreaks. And also, you can see that the outbreaks, they have not been confined to a single country or to a single time period. So Nipah virus basically, it shows a pattern of recurring outbreaks, particularly in Southeast Asia. From a regional laboratory perspective, I think it's important to understand that Nipah virus, it's not an isolated event. Therefore, it requires sustained preparedness and not ad hoc responses. So again, this map basically visualizes the number of Nipah virus cases reported over the last 2 decades, and you can see, although a lot of cases have been reported when we compare it to some of the other infectious diseases, but the alarming thing is that the high case fatality rate. And also you can see that the clustering of most of the cases is in the South East Asia region, again, which highlights that these countries, they share similar, ecological and epidemiological risks. Here, I just wanted to, shift the focus from human cases to the reservoir, which is food bats. We all know food bats, they are the host of Nipah virus.
Kemarin juga pas itu, cuman aku nggak sampein aja pas kemarin.
Can you please turn off? So important to realize that the geographic distribution of these fruit bats, it basically significantly overlaps with the region where Nipah virus cases have been reported. And what it means is that for public health preparedness, the public health laboratory point of view, the preparedness, it should be risk-based and not only about where the human cases have been reported. So I'll very quickly go through. We all know on January 26, India reported 2 cases of Nipah virus from West Bengal, and both cases were reported to healthcare workers. And this led to contact tracing of more than 190 cases, all of them which turned out negative. And we look at the public health response. So it included rapid case detection, isolation, contact tracing, enhanced IPC in healthcare facilities, and also they used the mobile BSL-3 lab. And WHO declared the risk overall as low. Similarly, on the 3rd of February, a case of Nipah virus, which was laboratory confirmed based on RT-PCR and ELISA, was reported to WHO by Bangladesh. And then this was basically this patient died on the 28th. And unlike the cases that were reported in India, this patient had, history of consuming raw date palm sap, which is a well-known risk factor in Bangladesh for Nipah virus. So again, different public health response activities were taken, and WHO has characterized the overall public health risk as low. So what we can learn from this? I think one of the key things is that it's important to realize that early diagnosis basically it helps in reducing panic, and it ensures efficient contact management. Also, it can reduce using extra resources or doing or implementing travel restrictions. Moreover, it's important that the primary cases they should not be missed, which can lead to delayed containment and also delayed contact tracing. And since this virus can cause healthcare associated transmission, therefore preventing transmission in healthcare setting is again very important. So it's a RNA virus, belongs to paramyxoviridae family, and you can see there are 6 different genes. Again, these genes they code 6 major structural proteins, and 2 of the major important proteins are the surface glycoproteins fusion and G proteins, which are important in entering into the host cell. So we know transmission could be direct from animals to humans, or it can be indirect, or it can be direct, sorry, from human to human through close contact with infected person. Clinical symptoms varies from respiratory symptoms, but important to understand that encephalitis is not the only clinical symptom. So there are other respiratory symptoms that we need to keep into mind. So there are case definitions for Nipah virus. And again, I need to highlight from the laboratory perspective, we need to ensure that whatever sample we receive are from cases that meet these case definition. Because we have seen during the COVID-19 pandemic that the labs, they get overwhelmed, and a lot of the samples, they do not meet case definition. They may be referred to the lab. So we need to keep that in mind. I think why labs, they are the first line of defense for Nipah virus. It's very important to understand because as I mentioned, the clinical symptoms, they can overlap with some of the other viral diseases. So it's important that we do the laboratory confirmation. And in case of Nipah virus, laboratory diagnosis plays very important role in early case confirmation, which can obviously lead to early isolation of the patient, and it can trigger infection control and contact tracing. And also laboratory confirmation, it supports outbreak detection, surveillance, and risk assessment. So again, I need to highlight that labs are the first place where panic can be prevented or it can be amplified. So we need to keep that in mind. So what are the diagnostics we have available for Nipah? So there are different diagnostics available, but molecular detection, especially using the real-time PCR, it serves as the gold standard for reverse transcription, real-time PCR. And normally the N gene is the target because it is conserved across the different strains. Our other gene segments can be used as secondary targets. And important to highlight the sample types as well. So for Nipah virus, throat sample, nasopharyngeal samples, and urine, they are ideal. Blood and CSF samples can also be tested in case of encephalitis cases. We have serological options as well, but they alone cannot be used for diagnosis. And because IgM antibodies in case of Nipah, they appear after 3 to 5 days. So they indicate recent or acute infection, active infection. And IgG, it appears after 2 to 3 weeks. So these are mainly used for retrospective diagnosis and serosurveys. And sequencing is also, a possibility, but it's not a diagnostic tool for Nipah virus at the moment, and it's mainly used in outbreak confirmation and genotype identifications. So this is a slide which shows, there are different, Nipah virus diagnostic kits, whether real-time PCR or ELISA, they are available on this website, which is the FindDX. I'm sure you have used it during the COVID pandemic. So you can scroll it using different filters, and it will give you the available CE-IVD options that are available for Nipah virus diagnosis. We also have different NGS or sequencing kits or reagents available. So I'll just quickly go through them. So there are some targeted or amplicon-based approaches that are already available, such as the viral surveillance panel from Illumina. Then NGI, they also have a commercially available solution to that. And then if there are, if you don't have and in targeted, then a metagenome approach can also be used. And one example is the NAB kit, which can amplify or sequence any viral RNA present in the sample. So there are two different genotypes of the Nipah virus. One is the Malaysian genotype, which is called NIVM, and then the Bangladesh genotype, NIVB. Both I think important to highlight that the transmission of the NIVM, the Malaysian genotype, is mainly pig to human, whereas the for Bangladesh genotype, it is from bat to humans or human to human. So, and again, the Bangladesh genotype has much higher case fatality rate, and the outbreak pattern, you can see it's mostly seasonal and recurrent. So this information is very important when we are doing surveillance for public health point of view. So in addition to diagnostic, I think key areas to highlight and to keep in mind whenever testing such samples in the lab is to make sure that you have the requisite biosafety requirements available. And again, this depends on the risk-based approach, whatever laboratory procedure you are using. And important to have restricted access, secure storage of the samples, and again, documentation. And lastly, it's important to have trained workforce that are properly supervised, people that are experienced in diagnosis, in basically, manipulating such high risk samples. So at virology department, we have different, labs, and I'll just highlight from in terms of Nipah virus, we use biosafety level 3 facility. We don't have a BSL-4 facility in Pakistan, but we use BSL-3 facility for testing such samples at virology lab at NIH. So we are the only reference lab in the country that can test Nipah virus at the moment. We have different facilities, infrastructure, and assays available, but for Nipah virus we use CDC validated real-time PCR assay, and for sequencing we also have the viral surveillance panel for Illumina, and if we want, we can also do metagenomics using NAB Next. So we have published different alerts of Nipah virus back in 2023 and just recently in 2026. This contains again important information on the Nipah virus and on case definition and how laboratory diagnosis can be done and how reporting takes place in Pakistan. So these are some of the steps that are followed for Nipah virus in Pakistan. So starting from detecting a suspected case to collecting samples and then referring those samples to NIH in triple packaging and maintaining the cold chain. At NIH, we test such samples using RT-PCR, and then we report to our Ministry of Health, whatever the results, and then to WHO and other relevant stakeholders. And based on that, different response activities are taken. And so all this, Nipah virus and other such high-risk pathogens, any activity related to this is monitored by our National Command and Operations Center. So, yeah, and there are several challenges in diagnosing Nipah virus in Pakistan. Although we do test these such samples at NIH, so we have limited diagnostic capacity, or you can say even no diagnostic capacity for Nipah virus in our provincial public health labs. So NIH only serves as the national reference lab where Nipah virus testing is done. So decentralization, I think it's a challenge. Biosafety is also a concern and challenge, as we just have very limited BSL facilities across our public health lab network. And if you want to do a molecular analysis or phylogenetic analysis, there is limited bioinformatic expertise, especially in our provincial public health labs. So, and again, in addition, there are some awareness and surveillance gaps, and also funding issues. So what are the key take-home messages, especially in terms of laboratory diagnosis of Nipah virus? I think one of the key take-home messages that early molecular testing, it saves times and it builds trust. And sample choice matters as much as the assay. So respiratory samples and urine samples, they are critical, especially for Nipah virus B outbreaks, which we have seen recently. And it's not important that every lab has the ability or the capacity to test Nipah virus, but lab triage is very important. I think it's important to that the peripheral labs they can initially point out or a suspected case, and then they can send those samples safely and securely to the reference labs in their countries. So serology and sequencing, they are not the diagnostic tool, but they complement PCR, and and they provide important information on the genomic diversity of the virus, which can be used in outbreak response. And importantly, it's important that whatever testing is being done for Nipah virus, it's done by trained staff and following all the biosafety and biosecurity protocols. So with this, I will end my presentation. I hope I have not gone over the time. Thank you.